Welcome to one of the most fascinating journeys in scientific history. This is the evolving story of the scientists, experiments and medical advancements that are decoding the mysteries of genetics. Gregor Mendel was a monk and scientist whose experiments with pea plants laid the foundation for modern genetics. By carefully tracking traits such as flower color and seed shape across generations, he discovered that inherited characteristics are passed from parents to offspring through discrete factors, now known as genes. Sickle cell disease is an inherited disorder that affects the gene that produces hemoglobin, the major protein that carries oxygen in red blood cells. In this condition, red blood cells are misshaped, which blocks blood flow to the rest of the body. The double helix structure of DNA was discovered in 1953 by James Watson and Francis Crick at Cambridge University, relying heavily on data produced by Rosalind Franklin and Maurice Wilkins. They deduced that DNA is a twisted ladder, or double helix, using X-ray crystallography images produced by Franklin. Dr. Robert Guthrie developed the first widely used newborn screening test in Massachusetts. The test detected phenylketonuria, a rare genetic disorder that can cause intellectual disability if untreated. Dr. Guthrie created a simple blood test using dried blood spots collected from a baby's heel prick. Following the passage of the National Genetic Diseases Act of 1976, the Genetic Services Program was established in 1978 within the Health Resources and Services Administration, specifically under the Maternal and Child Health Bureau. The Human Genome Project is an incredible scientific achievement. Launched in October 1990 and completed in April 2003, the Human Genome Project generated the first full sequence of the human genome. In response to the Human Genome Project, the CDC formed the Office of Genetics and Disease Prevention. The Advisory Committee on Heritable Disorders in Newborns and Children was a committee of experts that advised the Secretary of the U.S. Department of Health and Human Services about newborn and childhood screening for heritable conditions. ACHDNC became most famous for developing the recommended uniform screening panel. From 2004 to 2024, the National Coordinating Center for the Regional Genetics Networks worked with the 7 regional genetics networks and the National Genetics Education and Family Support Center to improve access to genetic services for underserved populations. The Recommended Uniform Screening Panel is a list of disorders that the Secretary of Health and Human Services recommends for states to screen as part of their state universal newborn screening programs. The RUSP includes a set of both core and secondary conditions. In 2014, the Centers for Disease Control and Prevention released Tier 1 genomic applications to identify genetic tests with strong scientific evidence and proven public health value. The initial priority diseases were hereditary breast and ovarian cancers, Lynch syndrome, and familial hypercholesterolemia. All of this is a large research program created by the National Institutes of Health with a mission to speed up health research so that everyone can obtain personalized healthcare to prevent and treat disease. In 2020, scientists at Johns Hopkins Applied Physics Laboratory and Johns Hopkins Hospital began using handheld DNA sequencers to rapidly sequence the genome of the virus that causes COVID-19. In 2023, the FDA approved the first gene therapies for treating sickle cell anemia. One therapy, Casgevy, uses CRISPR gene editing technology to modify a patient's blood stem cells to prevent the sickle shape. A second therapy, Lyfgenia, helps patients produce healthier hemoglobin. In 2024, the National Institutes of Health announced that researchers in the All of Us research program identified more than 275 million previously unknown genetic variants from nearly 250,000 whole genome sequences. About half of the data came from participants of non-European ancestry, helping address the long-standing lack of diversity in genetic research. In 2025, doctors at Children's Hospital of Philadelphia and the University of Pennsylvania became the first to successfully use a personalized CRISPR-based gene editing therapy to treat an infant with a rare and life-threatening genetic disorder called CPS1 deficiency. Researchers designed a custom treatment in just 6 months, using base editing technology to correct the baby's specific DNA mutation. We hope you enjoyed this journey through some of the most remarkable advances in scientific history. The code of life is still revealing its secrets, and the next breakthrough may be just around the corner.